thanks to what you learned in the material on the skeletal system, you already know that the dorsal cavity is going to protect both the brain and the spinal cord. So, in essence, the CNS. The cranial cavity protects the brain, and the spinal cord is going to be housed and protected by the vertebral cavity. So there is actually bony encasement of this very fragile nervous tissue in order to make sure that it is protected as possible. We will also have various layers of connective tissue, which are known as the meninges, which act to help anchor and protect the brain. There are 3 layers that you need to know. The most superficial layer is the dura mater, and dura for durable, mater for mother, and this basically says this is the tough mother. It is the outermost layer which actually attaches to the skull in various places, and therefore the brain is anchored so it's not slopping around when you move or if you smack your head. The arachnoid mater, or arachnoid, is where you will find a ton of blood vessels, and it is called arachnoid because it's kind of cobwebby or spiderwebby. Arachnoid stands for, you got it, spidery. Deep to that is the subarachnoid space, and a fluid, which is known as cerebrospinal fluid, will actually surround and bathe and move around the brain within the subarachnoid space. The deepest layer is the pia mater, and it is going to cover the brain and follow its contours just like a really tight sweater will follow the contours of whoever is wearing it. When you look at a dissected brain, you will see sort of this shiny covering of the nervous tissue, and that shiny material is actually the pia mater. Very thin, very delicate layer. So we know that the meninges are going to be membranes that are covering the brain as well as the spinal cord, and that shows you on the right the diagram of what they look like. So there's the dura mater, and it is going to be anchoring to the inside of the cranium in several areas. The arachnoid mater is this cobwebby area, and the pia mater, you can see quite clearly that it's going to follow the contours of the brain. Since we have this picture up, this shows you both white matter and gray matter, except it's sort of yellow matter and orange matter. So this material will be the white matter, myelinated fibers, specifically axons, and this material is supposed to be the gray matter, even though it's orange, and that will be unmyelinated axons. You have holes in your brain. I have holes in my brain. We all have holes in our brain, and they are known as the ventricles. The ventricles are going to contain cerebrospinal fluid, and that cerebrospinal fluid will be produced in what are known as the lateral ventricles and flow through the ventricles in the central canal and all around the brain. We'll talk about cerebrospinal fluid in a couple seconds. So they are going to be lined by what are known as ependymal cells. And we've already talked about ependymal cells. Ependymal cells are clustered together in the lateral ventricles in areas that are known as the choroid, choroid plexus. Plexus is a network. It's the ependymal cells that we talked about when we talked about neuroglia, specifically of the central nervous system. Is the ependymal cells that produce CSF. So the ventricles are going to be connected to one another and to the central canal of the spinal cord. They're going to be right and left lateral ventricle. They will be connected to the third ventricle, which is known, which is going to be, the connection point is known as the interventricular foramen. The third ventricle will be connected to the fourth ventricle through what is known as the cerebral aqueduct. So we know that the ventricles are within the brain. There are going to be 4 of them. There will be lateral ventricles, right and left lateral ventricles, one in each hemisphere. There will be separated by what's known as the septum pellucidum, which is basically just a connective tissue wall. The third ventricle is associated or located in the area of the diencephalon. Stay tuned on where that is. And the fourth ventricle is going to be associated with the hindbrain and basically just anterior to what's known as the cerebellum. So let's take a look at some pictures. Let's come here. So here are the lateral ventricles. This will be the right lateral ventricle. This will be the left lateral ventricle. Here's the third ventricle, which will be located mid-sagittally. The fourth ventricle is just going to be in the hindbrain and just anterior to the cerebellum, which is going to be this guy. And they will connect directly to the central canal, which is the hole that runs the length of the spinal cord. Now let's go back and talk about CSF. It's the fluid that is made by ependymal cells. Those ependymal cells are found in clusters known as the choroid plexus. They make about 400 or 500 milliliters of extracellular fluid, the cerebrospinal fluid, but much of that is reabsorbed, broken down, and reused. So the total volume of circulating cerebrospinal fluid, CSF, is about 125 to 150 milliliters. It is recycled about 3 times daily, so it's not the same stuff time after time after time. It is going to be recycled and replaced. This is what the cerebrospinal fluid does. It helps protect. It actually acts like a cushion, so it's going to protect the brain from damage because it sort of buffers the brain if you smack against something. Therefore, the impact of the blow or where you hit your head will be lessened. It also helps float the brain because it provides buoyancy. So we know that the brain is basically covered or immersed in fluid. The net weight of the brain is about 1,400 grams, and that is reduced to about 50 grams because it's floating. That means that the pressure that is at the base of the brain is significantly reduced, so that nervous tissue is protected because it is not actually carrying the entire weight of the brain. The CSF will also be flowing in a single direction. It's got a one-way flow. That means that harmful drugs or material, metabolites, are pulled away from the brain and put into the circulatory system. And it serves as a way that we can get hormones across or to various parts of the brain. So hormones that are released into the CSF will be carried to remote sites far away from where they were secreted, and they may act upon that area. It shows you the ventricles from a lateral viewpoint, and it tells you that the CSF will fill the ventricles, fill the central canal, the spinal cord, and surround the brain. Let's talk about the blood-brain barrier. What is it? It's a barrier between the blood and the nervous tissue of the brain. So we separate the blood that's in the circulatory system and brain tissue, as well as the CSF. We know that the CSF is produced and maintained by the choroid plexus. I keep saying that so you remember it. The choroid plexus is basically this network of ependymal cells. There will be specific areas within the brain where the cells of the blood vessels will actually restrict the diffusion of some of the material into the cerebrospinal fluid. What's prevented from entering? Pathogens like bacteria or viruses, large compounds, and certain hydrophilic molecules. Those cells that are restricting the diffusion of some of those materials will also allow the diffusion of other molecules: oxygen, hormones, carbon dioxide, alcohol, caffeine, nicotine, and barbiturates. So all of the things in that last line are not necessarily good, but yes, they can get into the brain and affect it. The cells of the blood-brain barrier are going to move metabolic products through active transport. So when you need to get glucose into a part of the brain, it will actually be moved by active transport across the barrier through carrier proteins. The blood-brain barrier can be broken down or damaged. And if you take a look at what can damage it, it's a whole list of things, many of which we are exposed to. How many of you know someone with high blood pressure, hypertension? Pretty much everybody. So yeah, high blood pressure can actually open the blood-brain barrier. Sometimes as the embryo develops into the fetus, the blood-brain barrier is not fully formed at birth. If you have a particularly high concentration of a certain substance, you can actually open the blood-brain barrier, and that substance can move into the brain tissue. Exposure to microwaves or radiation or certain infectious agents can open the blood-brain barrier. So those three elements can all impact the nervous tissue of your brain. Things like trauma, things like ischemia, things like inflammation, things like pressure are frequently associated with injury. So injury to the brain can actually open the blood-brain barrier. How do we prevent materials getting across the blood-brain barrier? Tight junctions. So there are actually physical junctions that prevent permeability to certain materials. There will also be metabolic barriers in the form of enzymes that will break down material that's not supposed to be entering the brain. Because of these, the blood-brain barrier is often what's known as the rate-limiting factor in the development of drugs, because how fast or whether or not they can actually permeate the blood-brain barrier is the rate-limiting factor in the development of different drugs. This shows you a regular capillary on the left. There might be small gaps, or there are also more small gaps, which are known as intercellular clefts between the simple squamous epithelial cells that allow movement of some material in and out of capillaries. When you look at the blood-brain barrier, you can see that there are tight junctions in between those endothelial cells, those simple squamous epithelial cells. In addition, the astrocytes, remember them, are going to have feet at the distal end of their processes. And those feet are going to basically lock together and add to the impermeability of the walls of the capillaries that are within the brain. The pericytes of the astrocyte, fancy way of saying the feet of the astrocytes, will also trigger the formation of tight junctions in between the capillary wall cells. Pretty cool. Now, there are several areas where the blood-brain barrier is weak, which means that there are substances that can cross the brain in those areas. So substances are going to be able to move across the brain in those areas because the BBB is weak. They're known as the circumventricular organs. And because of the circumventricular organs, the brain can actually monitor the composition of blood. There are multiple circumventricular organs, and they include the pineal body, what's known as the neurohypophysis, which is the posterior pituitary, and the area postrema. So let's talk about each of those. We'll mention them again. The pineal body, or the pineal gland, is an endocrine gland that is found in the brain, and we'll talk about that in another lecture, which secretes melatonin, not melanin, which is a pigment, but melatonin, which is a hormone. It's going to control sleep-wake cycles and your ability to adapt to varying amounts of day versus light, day and light or night. Pineal body will also secrete what are known as neuroactive peptides. The neurohypophysis is a fancy term for the posterior pituitary. The posterior pituitary is going to produce and secrete several hormones, including oxytocin and ADH or vasopressin into the blood. Another area where the blood-brain barrier is weak is the area postrema, and this is known as the vomiting center. So when you eat something that's bad or toxic, when that starts to enter the blood, it gets to the area postrema pretty quickly, and the area postrema will make you vomit because it is bad and it is trying to get you to expel the material from your GI tract. So that's the way that we can protect ourselves, by literally eliminating that toxin from your stomach before any more harm can be done. I love the area postrema. It's just fascinating to me that we have this control center which protects us from eating toxins. Fantastic.